down when you were inside on there. You might have to.
Yeah, this definitely seems like the place where it's at. There is a nice big bramble and this cool sea star.
Yeah, you're going to have to clear your verts.
Yeah, copy. Doing that now.
And Scott France has spotted a yellow coral just below this Paragorgia as well.
Clear? I think so, yep.
Okay. Yeah, it was verting down at 50% and barely moving.
Yeah. I think that
I think it's just at the bottom of the screen coming into view.
I think you got it.
We're dealing with a temporary thruster issue, watch lead.
Understood, pilot.
And is this the one that you were talking about, the one in view?
It's just off screen, right at the bottom middle.
Oh, copy.
You got enough tether, I can come down with you too.
Copy.
And it actually does not look
bottom
like that
middle,
cleared.
look at all those
okay.
acesta clam, there's scallops, and a bright yellow coral, and then there are all these really cool sea stars, the Brisingid sea stars, possibly in the genus Novodenia.
Okay, I think we resolved that temporary thruster issue. Watch lead.
Oh, perfect. Yeah, we'd like a zoom of that yellow coral first.
Copy. So, what do you think, co-pilot? Should I set up to the right of it?
I would maybe just try to hover on this one.
Copy. Will do.
You could put in auto depth now. It'll creep one way and then come right back.
Okay.
I mean, it's totally up to you. I think your Z bias is nailed down pretty well. But then once your auto depth sets, it's pretty locked in there.
Copy.
Yeah, then just adjust it as needed, and I don't think you have really any good places to set up on the wall.
Yeah.
Unfortunately.
Be kind of far away.
Yeah. Yeah, that auto depth is like super solid.
So we're coming to the end of our dive today at this box canyon that's located between Alvin Canyon and Block Canyon.
Gonna hold here.
Copy.
And this canyon is located within the Frank R. Lautenberg
I can get a little
Deep
closer.
Sea Coral Protection Area.
Yeah, it's right under Darcy's.
It looks like this is another Acanthogorgia yellow coral.
Good. Okay. You can go in more if you want.
Okay, coming in.
Yeah, that's like...
It just looks really fluffy.
That's cool.
And that's my gestalt to
okay,
Acanthogorgia, as they tend to look really fluffy, and then the polyps look very skinny and unable to pull in towards the skeleton of this coral. In the branches, I'm spotting a number of brittle stars and amphipods as associates.
Down a little bit.
And then all around this coral are these acesta flamed scallops.
Okay, rotating down a little bit more.
Some
I
with
should
encrusting
be.
Sponges.
Indeed, some do have encrusting sponges, and I saw a snail on another one.
Okay.
It's just a hot spot of life.
Heather's still rotating your head a little.
Okay.
But that's still, like, that's a great shot.
Mike White just knows how to pick them out.
Sometimes it works in your favor.
This has been a pretty cool dive.
Rotation.
There's also an anemone in the branch as well.
Yeah, I can't really ask for a better last dive.
Yeah, sorry about all those.
It's been pretty good.
I think the auto depth is still
lots of cephalopods, lots of corals, great collections. Hello.
Hey, this is Chris Ma.
Hey, Chris. You ready to see your really cool star?
Yes.
Okay, come back wide.
Okay.
Video, please.
It's a happy coincidence that because this area is
to your left toe.
so close to the lab of my Canadian colleague, that I actually have identified specimens of this species before, of the Brisingid that you're looking at. You are correct that this is indeed Novodenia, and the species is, appropriately enough, Novodenia americana. The animals have pedicelaria, little small claws, if you will, that if and when we get to zoom in on the spines, all of those club-shaped structures, those are actually spines covered with a sock full of these little kind of claw-shaped structures. And so those are what capture they can actually capture small crustaceans like little amphipods and other things. And when they're captured, they're actually moved by tube feet to the mouth. And so even though they're very graceful and flower-like, they're actually pretty big, deadly animals. And I've actually seen these. I think it was actually on one of these these North Atlantic dives. I've seen little cyclothony, those little fish, bristle-mouthed fish, run into the arms and get caught by the pedicelaria, and they don't. The one that we saw didn't have enough to get loose. So at least not for a while. So some of these brisingid can really even capture not just mobile prey but vertebrate prey. It's not clear if it moved away, if the fish got loose after the ROV moved on.
Okay, but you can go in video.
The brisingids are some of the first animals that I studied, and they're really kind of cool.
And is that lasers now?
The names are also really interesting because you were talking about how some of the names might actually reflect sort of their behavior. This one actually doesn't have anything to do with their functionality at all. These are all named after Norse gods, because the original one, Brisinga, is named after Brisingmen, a brooch that was thrown into the abyss by Loki. And so this used to be called, this genus used to be called Odinia, which was named for Odin, the god, the Thor's dad. And unfortunately, there's already a beetle named Odin, or Odinia, so it turned out they had to rename it. And so they named it New Odin, or Novodinia. So and these can always be recognized by that sort of really sort of club-shaped spine and
to zoom in
the sort of really big spiny structures that that you see on the disc and around the proximal part of the arm. That's the part of the arm that's next to the disc. So
should I come?
They're usually
back
more
you're
saltwater than where we are today.
I'm gonna hold
but but they get to be to be pretty deep. Yeah, you can see how very pronounced all of the spines are. That's very sort of classic Novodinia, although I think Novodinia antilensis is one that I typically see when we're looking at video from the tropical Atlantic. This is obviously farther north, and there's some question as to how closely related they are, or if they're possibly just the same thing. So, interesting animals. So, been fun
I think I can hold this one pretty good.
listening to the dive. Thanks very much.
Thank you, Chris. It's good to hear from you all the time about these really neat stars. I'm sad that this will be the last time you get to call in during our expedition.
Okay, coming down.
Well, I'll come back.
I know you will. You'll be on for the next expedition, telling us all about these sea stars.
Indeed. So have a good trip home and a smooth last dinner, and enjoy the first drink when you get ashore.
We will. Good night.
Coming out, pilot.
I slipped off. Thank you.
Bye.
So, should I try for more zooms on this one, Watchley?
Video is clear.
Thank you, video. Okay, I'll head over there.
Coming up. And rotators back to you.
Copy.
Yeah, just enjoying the probably the
can I
last
get
call in
10 minutes?
Deep
Copy,
Connections
10 minutes.
2019 from Chris
Watch
Ma.
lead nav. Yeah, about 10 minutes.
Invaluable
10 minutes left.
echinoderm consultant.
I think that reflection video
So now we're
is just
going
off
to
the
take
wall.
a good look at these bushy, I'm going to guess they're black corals here growing on the wall. That'll also be a new thing that we have not yet seen today or during any of our other dives.
I think you
And
can
as we
reach
zoom in,
that.
I'm going to look at the branching
up or
pattern
down
on
with
this
you.
coral, because that'll tell us a lot about what this might be. And also we have about 10 minutes left on bottom today.
Yeah, so you can
and I'm
see,
totally
like,
okay with not making to the top. This
really
is way
wants
cooler.
To rotate your head to the right. So
Okay.
it's just a constant rotation.
We just went back down.
So
yeah, because all the cool stuff is here.
Okay.
Along with everything else.
Yeah, but I'm not going to get that, FOMO like we did on the other dive at Beach Canyon where we just started to see the really cool stuff and had to leave. We saw some cool stuff, and now we're seeing even cooler stuff. Lots of cool stuff.
You're going to jinx it, and we're going to see the coolest stuff right before we come off bottom again.
That's totally how it works. But so looking at this black coral that we have in view, seeing these branching patterns coming off, and how that it's branching in a similar way to what a Bathypathes would do, but it has all these sub-branching on it. So I think this is a Teleopathes. And we'll see if someone's written anything in the chat. I see that Scott France has said that this must be Teleopathes magna. So it is pretty large, so magna does fit with this Teleopathes.
Yeah, definitely. It looks like kind of looks like a fern that you might see on land, but suspended on a canyon wall hundreds of meters above the axis.
I think I might just get a floating shot of this one. Now, what do you think, co-pilot? Should I just try and port tow back in?
No, I
float.
I think you could float. Yeah.
Okay.
Yeah, you can just do a snap zoom, pilot.
Copy. Okay, video, you can go in then.
Okay, coming in for partial.
Yeah, a nice good framing of this whole colony will make a really good image for our deep sea animal guide. And then I'm also seeing a cute little squat lobster with very long arms.
Nice. Look at those arms, man.
It's on the
right
side of the
spot
coral
lobster
pilot.
is a chirostylid squat lobster. They're usually characterized with these long, skinny arms. And we've seen them a number of times in black corals.
If it was just a little more flexible, it might have a future as an underwater wacky inflatable tube man.
Just ever so slightly.
Copy.
Nice.
Oh yeah, I can see that.
That's a very beautiful example of a black coral. This goes.
Can I zoom in a little bit?
So Scott's got some information about this coral that we're looking at. The holotype of this specimen, or this coral, was actually archived in the Nova Scotia Museum collection in Halifax, Nova Scotia, which is where we began our journey for this expedition.
Get polyps.
Yeah, that's taking us full circle. It's been quite an expedition. We started in Halifax.
A little less than 5 minutes.
We remained in Halifax while we had some weather and equipment related delays.
Nice.
We got some great dives in the Canadian waters, including the Gully Canyon Marine Protected Area, a couple of other Canadian canyons, transited into U.S. waters, went and hid from Hurricane Dorian for a couple days in Newport, got back out, made a beeline for the seamounts so we could explore both the seamounts and the submarine canyons that we were planning on exploring for this Deep Connections expedition. Yeah. Got back for a few canyons.
Video is clear, pilot.
Now
Copy that video.
Getting ready
coming
to
out.
head on home.
Thank you.
It's been a great 12 dives.
So Jeff, what would you say was your favorite thing that you've seen during these expeditions and these dives?
Got a crab coming.
Yeah, we've been putting together
highlights
and dive summaries and the report and all, so that kind of stuff is coming to mind. I don't know. It's hard to pick just one, but if I did, it might honestly just be Magnitudes Magna. I really like those things. Those are really cool.
I'm going to have to say my favorite thing was the Spongapalooza on Retriever Seamount.
Oh, man. Sponges beat out the corals. Don't tell them that.
There was just so many sponges. I just couldn't stop saying sponges.
I could not help but notice.
So if we could just end our dive looking at a sponge.
I am enjoying this crab rappelling down the very sheer rock face.
I'm really impressed with this crab's skill at holding onto the rock.
Truly impressive.
It really helps that it has all these small little holes. So, Pilot, I spotted a sponge actually down into the right.
Copy that. Got a little bit of time left.
Couple minutes left. Yep.
Copy.
Watch the nav.
There you go.
Go ahead, Nav.
Yeah, 2-minute warning.
Thanks. Just want a nice quick look at that sponge, and I
Copy
guess that'll
that.
be that.
I'll come down just a touch.
Copy.
So that was our 2-minute warning.
You can go in partial video.
Okay, coming in partial.
There's a little bivalve under there.
One last sponge for posterity.
With a
Holding
flame scallop wearing
I
that
do have more
sponge as
zoom.
a very fashionable bonnet, I think that would be called.
Yes, it does look like a bonnet. So fashionable. We have seen quite the deep sea
zoom in more,
fashion
Pilot?
throughout our dives.
Go ahead.
Okay.
Got the
So
headlights
many
video?
beautiful colors.
Sure.
So many beautiful shapes.
Might
The
help
great
with the underside there.
deep red pancake urchins come to mind. I think those win my deep sea fashion award for this this expedition.
Sorry, this current's...
It's okay. Here, let me zoom out just a little bit for framing.
Yep.
It is quite fashionable, so it's good to get a good shot of it.
There you go.
Okay, I'll
And
try
we've been
hold
seeing
that.
these
go.
small structures within the skeleton
Nice.
of this sponge, and they look like they might be reproductive structures, those little white dots throughout the sponge. And so we've actually preserved some of our specimens in formalin in order to preserve those structures for further study.
Yep, and all of this is
Okay, I was just
Okay.
kind of drifting
Coming
away
back to
because I
land
could keep it more
to,
stable.
That's fine.
hopefully answer some really
I'm gonna
neat
come out.
scientific
Copy.
questions in the future.
It was a good shot to end on.
Yeah,
So I
sounds
think we're
great.
doing
Yeah.
a slow
Headlights
pan
coming
out
off.
right now.
Copy, headlights off.
Just
That's
So I
to
could-
full wide.
I
get
think
our
maybe
last
I'll
look.
boogie it back up to the edge.
Okay, yeah, I'll come up with you as you do that.
Yeah, I was talking to Dive Super, and he said
What a dive, what
pretty
an
much
expedition.
if we just come up when you guys are ready,
It sure has been a
the
great
ship's just gonna
expedition,
stay
and
on
it
the
has
current
been a
heading,
wonderful dive.
come
I can
0.5
now
over
identify
ground.
more than
And
one
then,
biology.
when we get to about
thanks
500
to you.
meters,
You've
we're gonna
definitely
do
caught
0.5
on
through
with
the
a lot
water.
of the biology
So-
that you've been
Okay.
seeing,
That sounds good.
commenting even more than me some
And I
days
think-
on some of the
so,
I think,
biological
Pilot, all
aspects.
we have to do is
Just because I'm wrong on more than
For
half
me
of
to
them
come.
doesn't
port.
But the confidence is
Copy.
really good.
Okay, so
the
you go
effort,
port
A
to...
plus for effort. That's all
Yeah.
I want.
Yeah,
Yeah.
Yeah.
some of
And
the
then I would
best
go to starboard
biology
right.
by a
Right,
non-biologist I've
Okay.
heard in a long time.
Thanks, Megan.
So get a little bit up farther above this
Yeah,
wall.
and as
Yeah.
we're ascending
and I
above
guess we can
the
call it off the
lip
bottom.
of our
there.
box canyon
Yeah.
here,
Yeah, I would
Taking
just let
in
them clear
the
this
Okay.
connection
here.
Yeah, that's
between
fine.
the
I was just going to mark
continental
it.
shelf
Oh,
and
yeah, I
deep
think so,
sea
the
that
mark,
the
yeah, that's
submarine
right.
canyons
Just,
represent.
I thought you meant a call
I
bridge.
think this
No.
is
I'll just
a nice
I'll mark it.
opportunity for us to wish deep connections well.
I feel very deep and very connected.
Deeply connected.
So connected.
Okay,
So deep.
All right,
I just
that's
auto
delta.
depth in. It's going to servo up
Magnificent
a little bit.
connection.
Great. Yeah.
Connections to the surface water, connections to the deep sea, connections
Okay,
to Canada,
so-
connections to the U.S.,
So should I back up a little bit more, or
connections between canyons, connections between
I think
seamounts.
you're probably pretty good
Everything
there.
is
Okay.
connected.
It is.
I think, yeah, the easy way to do it
Everything
is
is
just for
connected
us
in the
both
deep
to
sea.
stay in
The deep
autos.
sea is connected to all of us.
Actually, you might want to back up because you're going to have to come 90,
So-
I guess.
okay?
With
Yeah.
that, I believe
Okay,
that
I'm coming back.
Copy
we are
that.
going to
Okay.
begin
I'll,
our ascent. So, with that, I would like to wish everyone
should I send
happy
her
rest of your weekend.
Come up just a
And please tune in for the next Okeanos expedition, which is going to be in November. Details forthcoming. And for the last time on Deep Connections, this is Jeff L2, Geology Elite, signing off.
All right.
And then one last piece of poetry before I sign off. There are no boundaries in the ocean. So we want to, we came here to study and explore and find out more about how the ocean is connected to all other locations within our oceans. And as stewards of the ocean, it's a responsibility to do these kinds of explorations. And I am so happy that all of you got to join us with these explorations, and I hope you had just as much fun as we have, because we've had a blast out here in the Northeast canyons and seamounts, and exploring all these new places that have never been seen before. So for the last time on this expedition, Deep Connections, this is Megan Putz, your biology lead from the University of Hawaii, signing off.
All right, that's close.
Yeah, and as we are leaving the seafloor, I just want to take a few moments to thank all the people that made this possible. So obviously it's a big team. We got close to 50 people on board, everyone that played a critical component to making this all happen. So, of course, the North Ship Okeanos Explorers officers, led by Commanding Officer Nicole Manning, Executive Officer Faith Knighton, Operations Officer Rosary Abbott, Joan Cachanago, Kevin Tarazona, Nico Osborne, and Brian Caldwell. I just want to thank the crew that they did an outstanding job at keeping the engines running, keeping the deck safe, and making sure that we could dive every day. Vincent Palazzolo, Rick Abona, Dave Blessing, Chris Ramley, William Royo, Peter Brill, Mike Collins, Nate Sapien, Jared Hasendorf, Chris LeBron, Pedro LeBron, Sydney Dunn, Ray Capati, Ellie Pacheco, James Scott, Celeste Mori, and Randy Collins. Also like to thank all the wonderful personnel from the Global Foundation of Ocean Exploration, our engineers that worked on the remotely operated vehicles, and the video engineers, led by Shan Kennison, Chris Ritter, Bobby Moore, Dave Wright, Mark Durbin, Chris Wright, Dan Rogers, Levi Onema, Andrew O'Brien, and Lars Murphy. Also, big thanks to our mapping team that was hard at work whenever we're not diving, led by Mike White. Also, big thanks to Dan Freitas and Charlie Wilkins. And of course, last but not least, I'd like to thank our science leads that did a terrific job. It was absolutely a delight listening to you folks, and I really appreciate all the hard work, narrowing the dives to 8 to 10 hours a day, and getting, processing the samples, and making sure everything is put away, writing reports. Also, big thanks to our sample data manager, Barry Eakins, who not only helped with sample data manager, but also helped streamline some of our forms. And then, of course, big thanks to everyone on shore. We had on this expedition 51 scientists and managers that participated regularly in dives. This included 51 people from 31 different institutions in 7 different countries and 13 different U.S. states.
Here in just a few minutes,
so big thanks to all of you.
Just let them line up.
Appreciate all the hard work over the last 3 weeks.
Okay, sounds good.
Thanks. Yeah, when you guys are ready, we'll start
moving. And thank
moving.
you, Dan, for coordinating a wonderful expedition.
Yeah, we'll get a little bit clear of this wall here.
Yeah, I figured you might want to get a little higher up.